Hi, and welcome to Workshop 35, featuring some of the top abstracts submitted into the ion and mucus transport category this year. My name is Anita Balázs. I'm a researcher at Charité University Clinic in Berlin, Germany. And I'm joined here today by my co-chair, Brian Button, Associate Professor at University of North Carolina. Brian will be working behind the scenes to manage the questions submitted to from our audience. I am very excited to hear everything from our presenters today about various insights on the physiological and pathophysiological aspects of airway epithelial ion transport, airway surface hydration, and mucociliary clearance. For housekeeping, please submit your questions to the Q&A. And without further ado, let's kick off our first presentation from Dr. Thomas Brett from Washington University. Dr. Brett will be talking about structural and functional insights into the regulation of a calcium-activated chloride channel, TMAM16A.
Hi, I'm Tom Brett at Washington University in St. Louis. And first of all, I'd like to thank Anita and Brian for inviting me to talk to you guys today about our work on the CLICA proteins, and in particular, how the CLICA1 VWA domain can potentiate TMAM16A anion channel in primary CF airway cells and tissues, and potentially how it enhances airway defense properties. And first of all, I have no disclosures to report. And so for a number of years, we've been working on the CLICA proteins, which are a set of calcium-activated chloride channel regulator proteins that when we started working on them were very not the mechanisms and functions of how they work or were not very well known, and they still really aren't. They've been loosely connected through genetic studies, SNP studies, overexpression type studies to a number of human diseases, including airway and gut diseases, because they're expressed in a lot of mucosal surfaces, mostly asthma, COPD, cystic fibrosis, some cancers, things like Crohn's disease. They were first discovered over 25 years ago. They were thought to be calcium-activated chloride channels themselves, but because they induce calcium-dependent chloride currents when you overexpress them in cells, but they don't have the structural features that are consistent with channels, and we and others have shown in the past that they are secreted multi-domain multifunctional proteins. And what we've been working on are structural, functional, and biochemical studies to elucidate the basic molecular mechanisms of CLICA function using structural, biochemical, and biophysical approaches, as well as cellular, and understand their role in human biology and disease, for example, in airway biology and airway diseases, in order to identify tools potentially that can target CLICA proteins to understand their function better, but also with the potential of developing therapeutics or to target or based on CLICA proteins. And in particular, today I'm going to talk to you about our work on CLICA1, the hallmark family member. It's been and in particular, applications of cystic fibrosis. And so I'm jumping right into it. Summarizing a lot of work, what we did with CLICA1, we found that CLICA1 is a potentiator for this calcium-activated chloride channel, which is also expressed in the airway, called TMAM16A. And CLICA1 contains, at the N-terminus, this matrix metalloprotease domain, which is responsible for cleaving the protein into two pieces. The N-terminal part of the protein can then bind TMAM16A via this protein-protein interaction domain, this VWA domain. And it can do that in a paracrine manner. In other words, you can just dump it onto cells and it'll meet up with TMAM16A and bind to it. And then what it does is it stabilizes TMAM16A on the surface, so that you get increased surface expression as TMAM16A is constantly recycling from the surface. It ends up there longer. You end up with more channels on the surface, therefore higher channel density, more currents. In other words, it doesn't gate TMAM16A. It stabilizes its surface expression. One thing I want to point out just briefly is that we determined the high resolution crystal structure of the CLICA1 VWA domain. Found that we observed that it was in a what we call an open configuration, which is a high affinity configuration for binding ligand. Just means that it can attain that. Doesn't mean that that's what the configuration that it's always in. And the interactions that we saw in the crystal mimic the way it will bind TMEM16A. In other words, there's a acidic residue from an adjacent protein binds to the metal ion-dependent adhesion site, and then this would mimic how CLICA VWA would bind to TMEM16A. This is all not all that important for the purposes of this talk, but the main point being that we can make milligram quantities, since we're able to crystallize the protein, we can make very pure milligram quantities of it for functional studies, such as this. In the model system that we used a lot, which was HEC293 T cells, where we take CLICA VWA domain protein, dump it on cells at a concentration of 5 micromolar, then do whole cell patch clamping experiments, and we can see that within minutes we see induction of the calcium dependent chloride currents through TMEM16A. Again, because we're impacting channel trafficking and not transcription, so it happens quite immediately. And through the years, we've from what we've learned from the basic science, our basic findings of how CLICA works biochemically, and structurally, there are a number of ways to target its function, potentially therapeutically. And what I want to, I'm going to focus on for the rest of the talk is this CLICA1 VWA domain, which is a protein-protein interaction domain, which binds to TMEM16A and potentiates it via the mechanism I just said. And what we wanted to look at was how, can CLICA1 VWA domain, can it potentiate TMEM16A in primary airway cells, and what are the impacts on airway mucus and defense properties? And in going forward, in going forward, I just want to point out before moving on that the CLICA situation in mice is fairly complicated. So there are a number of so for studying study human CLICA biology, CLICA knockout mice are poor, very poor models, because for one, CLICA1 is there are probably functional redundancies with mouse CLICA2, mouse CLICA4B, and there's a fair amount of literature out there with using mouse CLICA1 knockout cells or mouse CLICA1 knockout mice, which don't really show much in terms of phenotypes, and may not, or likely not reflective of at all of CLICA, what CLICA1 or any of the CLICAs do in humans. So in going forward, we wanted to study, what CLICA1 does in terms of human biology, human airway biology, and we focused on CF models because cystic fibrosis obviously is impaired by is hallmarked by impaired mucus and impaired mucociliary clearance in the absence of functional CFTR, unable to pass the major conduit for chloride and bicarbonate in mucus. You get thick, sticky mucus, which impairs mucociliary transport, and the lower pH of the mucus impairs the function of antimicrobial peptides like defensins and lactoferrins. The lower pH also alters mucin structure, and the advantage to studying CLICA TMEM16A synergy, how they can impact mucus properties, is that in this setting is that we can, in the absence of a dominant mucus modulator like CFTR, maybe we can better understand the potential of how CLICA1 TMEM16A can contribute to mucus properties. And also there's, the added sort of therapeutic investigation benefits that in the even in the era of effective CFTR correctors such as ivacaftor, elzacaftor, tezacaftor, there's still an unmet need to investigate how alternative channels can impact mucus properties that are ones that would that could possibly be supplements or independent of CFTR genotype. For one, there's, we have the patients that have class 1 mutations, which have early stop codons that cannot be treated with CFTR correctors. There's the problem of genetic background, that not every CF patient who has certain CFTR genotype responds the same to corrector drugs. There's potential for drug-drug interactions, and also, since these treatments have only been around for a couple of years, the long-term effectiveness is unknown. So, and so there's the need to, still the need, I believe, to, and others believe, to study, alternative channels in this setting. And in particular, I want to point out Enterprise Therapeutics 001, which has been shown in the last couple of years and been presented here at this meeting by Henry Danahay, and that is a nice selective potentiator of TMEM16A and has shown some good potential use. And in addition, such approaches could be applicable to other muco-obstructive diseases such as severe asthma, COPD, in terms of stimulating secretion to dissolve mucus plugs and that sort of thing. So, so we turned to CF systems, in particular primary CF airway cells and tissues, to investigate CLICA1 TMEM16A function in terms of in mucus biology. And so, first of all, what we had to demonstrate or had to investigate was, can CLICA1 actually potentiate TMEM16A in primary CF airway cells? And what we did was, from explanted lungs, harvest and epithelial cells, culture them. This is in submerged cultures. We first had to just demonstrate by simple patch clamp experiments in submerged cells that we could potentiate TMEM16A. And what we see is in a number, in CF cells from primary CF cells from a number of class dysfunction classes, including class 1, we see that either full CLICA1 applied to can potentiate TMEM16A, or even just the VWA domain, and these currents are blocked by TMEM16A. So we can potentiate TMEM16A in this setting using, and this is just showing reproducibility, and then we block these currents using TMEM16A inhibitor, T16A01. Then we had to move on to looking at monolayers, epithelial, look at transepithelial currents in airway, well-differentiated airway cell monolayers. Again, we apply CLICA1 VWA to the apical side, and then do Usine chamber experiments. And what we saw in these experiments, at least with this this class 2, class 1 CFTR class combo, we saw increased baseline currents. And then also with UTP stimulation, we see an increase that the currents are actually more stable. They degrade slower. In another sample in DEL-F508 homozygous, what we saw were that CLICA1 VWA application increased the UTP-dependent currents through TMEM16A, and then also that increases the baseline currents again, and also the UTP-stimulated currents. In this particular sample, we did not see the increase in stability. So next the next thing we wanted to look at was since we've shown that we can potentiate TMEM16A using CLICA1 VWA in primary CF airway cells. Next thing was to look at, does it impact mucus properties? And so we looked at staining of CF tissues, and we noticed that that, like this example here, we could see TMEM16A expressed in the airway epithelium and what looks like secretory cells, possibly club cells, and also in the inner linings of submucosal glands. So we wanted to capture that, and not just, so instead of just using ALI cultures, what we went to decide to use for these assays was tissues from explanted CF lungs, which was fairly challenging. So what we did was take use either mainstem bronchi from or large airways from explanted CF lungs, and from normal donors use tracheas, and we could put them in culture for like 4 to 5 days. So we had to do these experiments pretty quickly. We would treat them with CLICA1 VWA, 5 micromolar, or with just the vehicle, with just buffer. We would always have CFSE stain in there so that we could see the areas on the tissue where we applied treatment. And then we would, using 2-photon microscopy and 1 micrometer fluorescent beads, 1 micrometer fluorescent beads, we would take short movies of CFSE stained areas, 3 movies per area, 3 areas per tissue, about 4 tissues per patient. And then to ensure that we were looking at mucociliary transport flow due to mucociliary transport, we would quench using peroxide, which kills ciliary bead. And what we saw, I'm sorry. And then to date, we've been able to look at samples from 4 patients, a couple of del F508 homozygous, and then excitingly, a couple of class 1 severe, class 1 mutations. And what we saw was in donor, you can see nice movement of beads with mucociliary transport. We had click it to that. We don't really see any improvement in this particular sample. We actually see some slowdown, but usually it didn't do anything. And with the class 1, we see before treatment, we would see beads just sitting there, stuck in the non-moving thick mucus. And if we applied CLICA1 VWA, then we see this rapid increase, dramatic increase of currents, or I'm sorry, not currents, but of bead flow, which was close to what we observed in healthy donor. And that's just shown here, what we saw for both of these samples, that we see this dramatic increase in bead velocity that was about 80% of what of that observed in healthy donors. And so in summary, what I've told you is that CLICA1 VWA binds TMM16A and can stabilize that channel on the surface and increases channel density to increase currents. CLICA1 VWA can selectively potentiate TMM16A in human primary CF airway cells, and this potentiation enhances mucociliary transport in CF airways. In future directions, we'd like to investigate how it impacts other mucus properties such as ASL thickness and mucus pH, and potentially investigate other CLICA TMM16A family members, in particular TMM16B, which is also expressed in the lung, although we have to investigate exactly where. We haven't figured that out yet because we have to find new antibodies. But we recently showed that CLICA4 VWA domain, another family member, specifically binds and potentiates TMM16B. And so along those lines, could we potentially harness that channel to impact airway biology? And in closing, I'd like to thank my collaborators, Colin Nichols, Electrophysiology, and Jen Alexander Brett, who did the lung tissue, cell and tissue culture, and Mark Miller, who does the 2-photon microscopy. And this work was mainly done by Kayla, Zainab, and Monica, and all of our funding sources. And thank you for listening, and I'll take questions.
Thank you, Dr. Brett, for this very insightful presentation. One of the first questions was about the basal current that you observed in Using chamber. So what do you think this could represent in absence of an exogenous purinergic stimulation? And they ask why you don't see that in normal donors, but you do see that increased basal current in CF cells?
Oh yeah, thanks. That's a good question. We actually, so in terms of like the increased baseline currents, maybe it's just, we don't really know actually. Like there is increased, we think CLICA is increasing the TMM16A, channel density on the surface, but and maybe there's some leaky currents due to that, just low basal currents without, purinergic stimulation. And maybe that's what it is, but we don't really know. And in terms of I'm sorry, what was the other question? It had to do with.
So why you didn't see it in healthy donors?
In normal donor, yeah, I forgot about that. That we're not quite sure of either, why we don't, but it's consistent that, I mean, and in the normal donors, the TMM16A expression level is much lower. When we get the cells from the CF lung, they've been under, inflammatory signals for a while, and the TMM16A expression is pretty high. So it could be due to that, but.
Then another question about the variability of response. So why was it quite variable in CF cells, some having large responses and others having little or no effect?
Yeah, I mean, we usually saw the same trends. It's just like whether or not the magnitudes were very much. And again, I would just speculate that it probably has to do with the TMM16A expression levels that were in, given cells. But that's about all I know.
Another question, why was IL-13 necessary to see an effect of CLICA1 in well-differentiated cultures?
Right. So with those, I think there you're talking about the Leucine chamber experiments. With IL-13, I think we did have to add that to get some TMM16A expression in those, just because they'd been, in culture for quite a while. So to get them to go to well-differentiated ALI. And so we had IL-13 to increase the TMM16A expression. Yeah, so.
Let me ask you one question which I was interested in. So there was this back and forth debate on whether or not it is beneficial to stimulate TMM16A because it could be also inducing mucin release, mucus secretion. And for instance, what are your thoughts on it? Or do you see in your explant cultures evidence of mucus release when you applied your CLICA1 subunit?
We didn't see any evidence of that, no, when we, not stimulating, so we didn't see any evidence. We didn't look for it specifically, but we didn't see any sort of evidence of that we were stimulating mucus secretion. And I think that's been shown by Henry Danae and others that, for their potentiator, their small molecule potentiator, TMM16A, that it doesn't. But yeah.
And another question also along these lines, if you saw any smooth muscle reaction in like that would be not that would be counterproductive when you apply this CLICA1 treatment.
Yeah, we didn't have smooth muscle in there, so we didn't, we that was gone. So I don't think so.
Then another question, Kyle Kunzelman has suggested that in addition to stimulating ion secretion, CLICA1 is a potent stimulator of mucus and secretion and production. Have you looked at mucus production in your explant cultures with exogenously added CLICA1? Yeah, we touched on this already, right. But maybe you could expand.
Yeah, like I said, we haven't noticed that, but we haven't looked specifically for it. But like I said, I think I thought, from what I remember, I thought Henry Danae had shown that with their EXT 01 potentiator, their small molecule, that doesn't happen.
Have you tried the CLICA1 in addition to CFTR modulators?
No, we have not tried that experiment yet, actually. Yeah, it's been on the docket, but we just haven't done it.
From a structural point of view, now you studied this interaction between TMEM16A and the CLICA1. Do you think that you could now design a small molecule to mimic this interaction, or would you need this protein subunit? And how do you envisage that as a therapy for CF?
Right, that's what we're trying to get at. So we're trying to get towards that, we're trying to get a structure of the complex of TMEM16A with the CLICA, with CLICA1 VWA, or the full CLICA and part of CLICA that binds to TMEM16A by cryo-EM, and we have some data, and we just have to work through that. But and then from there, maybe we can get some clues as to how we can make like a peptide mimic or some kind of thing like that could do that. And but yeah, those are some of the goals, but we aren't there.
And since you showed that the CLICA1 is expressed by secretory cells, so how do you think that happens? Dysregulation in vivo, in a physiological setting. So would it be secreted, and in a paracrine fashion it reaches ciliated cells where chloride secretion mostly happens by TMEM16A, or what would be the mechanism?
Yeah, no, I think it definitely could, like in vivo, like that's, like whatever, and in actual tissue, that's probably how it does happen, like is able to be secreted by, whatever. If the expression are on different cells, yeah, it could be expressed from one cell, float around in the milieu, and end up, potentiating channel on another cell. That's basically what we've seen in culture and co-cultures and kind of experiments. We've done those kinds of experiments all different kinds of ways, just dumping protein on, or just co-expressing, like whatever. Yeah, just doing co-culture kinds of experiments. So yeah, I think that's exactly what happens.
Okay, I think our time is up. Thank you very much for the lively discussion. And then we should move on to our next presenter, Dr. Yu Mikami from UNC Chapel Hill, and he will talk about and present new data on mucus obstruction, hypoxia, and sodium reabsorption.
Hello, everyone. I'm Yu Mikami, a postdoc at UNC at Chapel Hill in Dr. Boucher's lab. First of all, thank you all for giving me this opportunity to make a presentation at this workshop session. Today, I'd like to talk about airway obstruction produces hypoxia-dependent sodium absorption in human airway epithelial cells. There are no relationships to disclose related to this presentation. CF lung disease affects the conducting airways and is characterized by airway mucus obstruction. This is an airway in the CF lung with ABPA staining. As you can see, airway epithelium is covered with thick mucus. CF mucus is hypoxic. This evidence was obtained by oxygen microelectrode measurements in vivo in the proximal airways of CF subjects. Airway epithelial cell can gain oxygen from luminal air, and if the airway epithelial cells are covered with thick mucus, the airway epithelial cells may become hypoxic. The studies with hypoxia probe in vivo revealed that the airway epithelial cells underneath mucus plugs were hypoxic in the beta-inoc mice model of muco-obstruction. Previous research showed that short-time hypoxia attenuated ion transport in rat alveolar epithelial cells. Chronic hypoxia on cells energetics and functions differ dramatically from acute hypoxic effects by cancer immune cell study. Because chronic hypoxia has the potential to alter the disease course once it is established in the CF lung. It is important to understand the relationship of hypoxia to mechanisms that regulate airway surface liquid and mucus function. But the effect of chronic hypoxia on HB cells is still unknown. We hypothesize that human bronchial epithelial cells can adapt to chronic hypoxia, and chronic hypoxia changes ion transport on HB cells. Thus, this study aimed to evaluate CFTR and ENaC ion channel activity in well-differentiated HB culture under chronic hypoxia. Primary HB cells were cultured on transfer utilizing the ALI culture method for at least 3 weeks until the cells were well differentiated. Then the cells were cultured in a hypoxic chamber with 1% O2. This is our hypoxic chamber. This can control O2 concentration, and the cells culture box inside the chamber can keep the humidity and temperature like a regular cell culture incubator. We can keep culturing the cells and collect samples without exposing the samples to ambient air. For the oozing experiment, we developed an oxygen controllable oozing chamber for this study. Okay, let's move on to the results. HB cells were studied under 3 different conditions: normoxia, cultured under normoxia, and placed acutely into an hypoxic oozing chamber as a model of acute hypoxia. Cultured under chronic hypoxia for 5 days and studied 1% O2 oozing chamber. This is a chronic hypoxia model. The acutely hypoxic cells showed significantly lower basal short circuit current, reflecting decreased delta amyloid sensitive current and phosphorin stimulated current compared to normoxic cells. These results are consistent with previous reports that acute hypoxia attenuated ion transport. Unexpectedly, HB cultures exposed to chronic hypoxic conditions showed significantly higher basal and a large increase in delta amyloid sensitive current relative to normoxic cells. CFTR remained functional and mediated chloride secretion at rate similar to normoxic levels under chronic hypoxia. Thus, the cells appear to change and adapt to chronic hypoxia, resulting in upregulation of ENaC activity. We next investigate the molecular mechanisms mediating increased sodium absorption by measuring the gene expression of ENaC subunits in normoxic versus hypoxic HB cultures. As you can see, the chronic hypoxic HB cultures showed significantly upregulation of gamma ENaC and beta ENaC, but not alpha ENaC mRNA compared to normoxic cells. This is a Western blotting images. Surface gamma ENaC expression was also significantly upregulated in hypoxic cells. The percentage of solids in the apical secretion was higher in the chronic hypoxic culture, reflecting the liquid absorption. The next slide shows the studies of gamma ENaC mRNA expression HB culture and individual HB cells using the single-cell RNAscope. You can see the upregulation of gamma ENaC mRNA on hypoxic HB culture and the increase of cells that express high levels of gamma ENaC in hypoxia-exposed cells. This is kind of the gamma ENaC hot cell. We then evaluated if there was evidence that gamma-INAC mRNA was upregulated in vivo. This is gamma-INAC RNA-scope results for normoxia, normal, and CF airways. You can appreciate gamma-INAC mRNA expression level was by RNA-scope was significantly upregulated in the mucus obstructive CF compared with control airways. I'd like to talk a little about hypoxia-inducible factor, HIF. HIF transcriptional factors are master regulators of the cellular response to hypoxia and coordinate a transcriptional program that ensures optimal function, metabolic, and vascular adaptation to O2 shortages. When oxygen tension drops, PHD and FIH activity are inhibited, leading to HIF-alpha accumulation and nuclear translocation, heterodimerization with HIF-1 and recruitment of P300/CBP. The HIF transcriptional complex binds to hypoxia response element, HRE, to control target gene expression. We evaluated if the knockout of the gamma-INAC and HIF1 gene changes to the upregulation of sodium absorption on chronic hypoxic culture. Gamma-INAC gene knockout by CRISPR-Cas9 technology attenuated upregulation of delta amyloid sensitive current in hypoxic culture. HIF-alpha knockout restored the upregulation of gamma-INAC mRNA by chronic hypoxia. HIF-alpha knockout also attenuated the upregulation of delta amyloid sensitive current by chronic hypoxia. Therefore, HIF-alpha may play an important role in this process. To summarize, chronic hypoxia in culture promoted sodium absorption through the upregulation of gamma-INAC. This seems to be a feature of muco-obstructive region of CF airways. HIF-alpha knockout restored the upregulation of gamma-INAC mRNA and delta amyloid sensitive current by chronic hypoxia. So, chronic hypoxia induced gamma-INAC mRNA through the HIF-alpha pathway and promoted sodium and water absorption from airway surface and induced hyperconcentrated mucus. Thus, we hypothesize that chronic hypoxia may induce positive feedback cycle that perpetuates mucus accumulation in the CF lung. The restoration of airway normoxia would be the therapeutic strategy for CF lung disease. I want to thank my lab members and collaborators who supported me. This study was supported by the Cystic Fibrosis Foundation. Thank you very much for your attention.
Thank you, Dr. Mikami, for this impressive data set. One of the first questions was, have you characterized which cells are expressing high INAC, I assume in response to hypoxia?
Thank you for the question. So can you hear me? This is really great question. I also run the single-cell RNA sequence to characterize which cells are expressing INAC. As you know, INAC are consistent with the alpha to alpha, beta, gamma, delta INAC. So as far as I analyze the single-cell RNA sequence data, alpha INAC mRNA broadly expressing in the variety of cell types, and beta and gamma ENaC expression tend to be higher in the basal and suprabasal and secretory cells. So, and also the gamma ENaC hot cell could be in the part of the secretory cells. So this is the answer of the question.
Thank you very much. Next question is if you applied a bilateral hypoxia, or have you tested only apical or basolateral hypoxia, the effect of only apical or basolateral hypoxia as well?
Thank you very much. That's really great question. Actually, in the presentation of this slide on this study, I use a bilateral hypoxia, but I also developed a kind of special system making the asymmetrical hypoxia, which can develop the hypoxia only in apical side. We also check the gamma ENaC expression in such HB cell in such culture condition. Even asymmetrical hypoxia upregulates gamma ENaC in HB culture.
Thank you very much for the answer. Another question relates to the specificity of hypoxia on the effect of other ion transporters. So you demonstrated that hypoxia doesn't have an effect on CFTR function. What about calcium activated chloride conductance?
Thank you for the question. Actually, I didn't show the result of the data, but I think there are not big difference in the CSCC activity in both in chronic hypoxia.
Another very interesting question: if you try to reverse hypoxia, and did the cultures return to normal in a high oxygen environment?
Yeah, that's really important question. So actually, this is now ongoing study, so I can say the correct answer. But I hope the returning to the normoxic oxygen level could be restored the upregulated ENaC level.
Thank you. So you presented this self-perpetuating vicious cycle of mucus plugging, hypoxia, enhanced sodium reabsorption, and of course, how to break this cycle to restore ventilation, but that's easier said than done in a CF lung. So how could you target this this cycle to break it?
Okay. Yeah, this is important point for the therapeutic, and I also mentioned the end of the slide. So maybe your question could be how to restore the hypoxic airway to normoxic. So my idea is consistent with two. One is remove the mucus physically using the mucolytic reagent. And second one could be using the HIF-1 alpha inhibitor or kind of the hypoxic pathway inhibitors. So if we can use such kind of the reagent without not strong side effects, it could be really good therapeutic option for CF, and not only CF but also other muco-obstructive lung disease patients.
Thank you. Another question: is airway hypoxia as a result of thick mucus layers or mucus plugs where air cannot diffuse more distal, or both? So is it the plug, the airway, or is it just the mucus layer and underneath the epithelium cannot get enough oxygen?
Yeah, that's really great question. Yeah, actually, to be honest, I don't have the exact answer, but the previous study said mucus plugged epithelial surely under hypoxia, but the study didn't show the distal side of the airway condition. Aloe vera has a kind of the pore between the airways, so maybe some air might be diffused to the distal side of the airway, but I don't have the correct answer of that.
One question from my side is it's been shown that in the beta ENaC mouse that hypoxia can induce necrotic cell death and release of IL1 alpha. And I wonder if you have tested that in your system.
Actually, I didn't run the IL1 alpha treatment on the hypoxic cells, but that's really great point. So in the near future, I definitely do such an experiment. Thank you.
One idea maybe, have you seen mucus accumulation in the hypoxic cultures? So that would be maybe an indication of the IL-1 receptor activation and mucus secretion through that pathway.
I have actually I have checked the apical surface. Percent solid of the apical surface, the percent solid surely increase, but the exact at least MAC5B, exact MAC5B secretion did not increase under the hypoxia. So the mucus accumulation is majorly due to the activation of the ENaC.
I see. Thank you very much. And you already touched on it, but maybe you can expand. Similar question relating to how to interrupt the vicious circle in non-CF conditions. For instance, in a respiratory infection where there is transient hypoxia present, how to break the cycle.
Yeah, actually, that's really important question. And also, as I answered before, maybe mucolytic reagent or kind of the inhibitors to block the hypoxic signaling could be really important for treatment for the such patients.
Thank you. Maybe one last question. Are there any HIF1 inhibitors in clinical use or in clinical development?
Actually, there are tons of the HIF1 alpha inhibitors, but I think there are not so specified HIF1 alpha inhibitors. So I use several HIF1 alpha inhibitors. One of the inhibitors worked well, but other didn't work. So yeah, maybe such kind of the inhibitor still under development phase.
Thank you, Dr. Mikami. And then our time is up. Let's move to our next presenter, Alex Harris from University of Alabama, and he will talk about interesting role of mucin sialylation.
Hey everyone, I'm Alex Harris. I'm a PhD candidate at the University of Alabama at Birmingham in the labs of Dr. Stephen Rowe and Dr. Jared Barnes. And I'd like to first and foremost say thank you for the invitation to present my work to you all. It's quite an honor to present to and among such an esteemed group of scientists. And I'm excited to share my work. So the title of my talk is Altered Mucin Saliation Results in Delayed Mucociliary Transport and Cystic Fibrosis. So I have no relationships or conflicts of interest to disclose related to this presentation. And unfortunately, Alexicaptor wasn't named after me, and that's just a funny coincidence. So I thought I'd clear that up. As most of you already know, cystic fibrosis, or CF, is a genetic disease caused by mutations in the CFTR, or the cystic fibrosis transmembrane conductance regulator gene. And CFTR is a transmembrane anion channel on the apical surface of the epithelium, which is responsible for the secretion of chloride and bicarbonate across the apical cell surface. and it's essential for maintaining proper hydration, pH, and mucus properties in multiple organs. And the organ that I'm going to be focusing on today is the lungs. And in the lungs, impaired CFTR ion transport results in abnormally adherent and viscous mucus in the airway, leading to mucostasis. And it's well known that airway dehydration and mucus hyperconcentration reduce mucus clearance, and that altered mucus biogenesis via the abnormal electrostatic environment also contributes to CF mucostasis. And together, this pathophysiology paves the way for chronic infection and lung damage that leads to lung failure and early mortality. And showing this in this figure from Jeffrey Wan and colleagues is a CF lung explant. And you can see this mucus plug here and how dehydrated it is and how stuck it is to the airway. And this is what causes that chronic inflammation, lung damage, and provides a perfect niche for chronic infection in patients affected. So it's well known that dehydration is a major contributor to mucostasis in CF, and recent evidence has shown that mechanisms beyond dehydration also contribute to CF mucostasis. And highlighting this in this figure from Hager and colleagues, where they took CF or non-CF and CF piglet trachea, and they stretched it out across this panel. And they stimulated on the right side here with methylcholine, which simulates mucus secretions as well as ciliary beating. And you can see in the non-CF group, when they stimulated with methylcholine, the trachea were able to clear the mucus no problem. But in CF, they weren't able to clear this mucus and it remained stuck. And even when they submerged this in saline and provided adequate hydration, it still remained stuck, arguing that hydration alone wasn't enough to correct this CF mucus defect. Furthermore, in order to recapitulate the CF mucus defect in a non-CF model, they had to simultaneously deplete bicarbonate and chloride, and neither alone were sufficient enough to induce the CF mucus like stasis. And together, this just shows that other mechanisms beyond hydration contribute to CF mucus stasis and may be therapeutically relevant. So what is mucus? So mucins are the primary constituents of mucus that are responsible for many of its gel-forming properties. They're large 5 to 50 megadalton glycoproteins that are heavily oligocosylated, with carbohydrates contributing up to 80% of the total mass. And among these carbohydrates is heavy terminal salivation, which is provided by salivtransferases throughout the Golgi network. And this salivation provides the majority of the negative charges on mucins. And these negative charges are extremely important for interactions between mucins and their ionic environment. And to demonstrate this, in this figure by Kessler and colleagues, shows how mucins are packed within the cell. And they're packed in calcium-rich granules. And this positively charged calcium helps stabilize all these charges coating the mucins. And upon secretion, these calciums are exchanged for double the number of sodium ions, which forces these mucins to take up water and expand into a linear conformation where they are carried up and out of the airway. And most of the work to this day has been focused on the ion part, or the ion dysregulation in this ion-mucin interaction. And I'm more interested in the intrinsic electrostatic properties of mucins, i.e. their salivation state. So the main question we're asking with the study is, does negatively charged salivation of gel-forming mucins alter the physiochemical characteristics of mucus? And so throughout the years, it's been shown that terminal salivation is altered in airway cells with impaired CFTR anion transport, as well as mucins with less negative charge have been correlated to a more globular pathologic mucin conformation. And these lower charged mucin glycoforms have also been implicated in multiple diseases of pathologic mucus, such as COPD and asthma. But to this day, the effect of impaired CFTR anion transport of mucin salivation still remains unclear. And the link between altered salivation and mucus charge physiology in CF hasn't been determined. And this together leads me to my overarching hypothesis, which is that impaired CFTR ion transport downregulates the salivation of mucins, which decreases their negative charge and contributes to mucostasis in CF. And I plan on testing this hypothesis with two primary aims. Aim one, to define the effect of defective CFTR ion transport on mucin salivation, and aim two, to define the consequences of reduced salivation on the physiology of secreted mucins. And so the first question we ask is salivation reduced in CF? And the first thing we wanted to do was see if there were any differences in salivtransferase gene expression between CF and non-CF of salivtransferases that may be, or that are thought to be involved in the O-link salivation of our mucins. And so we took our primary human bronchial epithelial cell model, differentiated and cultured at air-liquid interface, and we took cells from non-CF and CF donors, and cracked them open and performed a targeted qPCR array looking at our solid transferases that may be involved in O-linked silation of mucins. Interestingly, we found that 4 out of 5 of these were downregulated, and 2 of these, almost 3 of these, were significantly downregulated. And out of the genes that were significantly downregulated, they represent the ST3-gal and ST6-gal family, which are the 2 primary solid transferases that add O-linked saliation to mucins in an alpha-2-3 and an alpha-2-6 fashion. And this together just shows that, or provides evidence that saliation may be down in CF. And so then we wanted to look at the secreted mucin itself and determine if there were any changes in secreted mucin saliation between non-CF and CF, and if this had a relationship to CFTR and ion transport. And so we took, once again, our primary epithelial cell model. We took our CF cells and non-CF cells and treated with our CF cells with either vehicle or elexacaftor, tezacaftor, ivacaftor modulator combo to correct CFTR and restore anion transport, or we treated non-CF cells with just vehicle. And first, we wanted to assess mucus transport to confirm that our CF and non-CF models and our CF-directed model do have the proper mucociliary transport physiology before we looked at their salivation state. And to do this, we use micro-optical coherence tomography, or micro-OCT, which is a technique used heavily by our lab. And to orient you to these videos here, it allows us to take cross-sectional live videos of cell monolayers as well as trachea. And so what we're looking at is this bright part here is the filter the cells are grown on. This is the epithelial monolayer, and denoted by this yellow bar here is the air surface liquid, or ASL, and this is where the secreted mucus is. As you can see here, our CF vehicle, the mucus is static. Mucus is moving in a normal fashion in our non-CF vehicle, and we were able to restore about 40% of our mucus transport in our CF model by treating with this elexacaftor, tezacaftor, ivacaftor combo. And I have this quantified here. And so now that we established that our model had correct mucus transport, we wanted to see, what is the salivation difference between the mucus from these cells. So we collected the secreted mucus from these Samuelside filters, partially purified them for mucins, and subjected them to lectin blots with SNA that recognizes alpha-2-6 salivation and MAL2 that recognizes alpha-2-3 salivation. And interestingly, what we found, similar to our mucus transport findings, was that for alpha-2-6 and alpha-2-3, our CF groups had less salivation than our non-CF groups, and this was significantly restored with corrected CFTR with our triple modulator combo, which once again provides evidence that salivation may be down in CF, and this may be directly linked to defective CFTR. And so next we asked what happens to mucus transport when mucin salivation is reduced. And so we wanted to see if we could directly reduce the salivation of these secreted mucins so we could assess what happens to their mucociliary physiology. And to do this, we implemented a tool, a salivary transferase inhibitor, 3-FAX-paracetyl mu5AC, which I will term SCI from throughout the rest of this talk. And it is a salic acid analog that competitively inhibits salivary transferase, preventing the addition of salic acid from a donor, from its donor to its sialicide, which in our case would be our mucins. So we wanted to see, could we indeed decrease the salivation of our secreted mucins in our cell model? So we took our STI, treated basolaterally to our non-CF cells, waited 24 hours for mucus to be secreted under these conditions, and then we collected the secreted mucus and subjected it once again to lectin blots to probe for salic acid. And in this case, we used free germ agglutinin, or WGA, which recognizes the overall salivation abundance on our mucins. And as you can see in our slot plot, which is quantified here, there is significantly less salivation of mucins after treating them with this STI. And we also confirmed this using agarose page, where you have 3 different volumes of mucus added for our control and STI, and you can see that there's a drastic reduction in salic acid staining after STI treatment. And so now that we confirmed that we were able to indeed decrease the salivation of our secreted mucins, we asked, what is this doing to mucociliary physiology? And so once again, we took our STI, treated our non-CF cells, waited 24 hours for mucus to be secreted, and then assessed mucociliary physiology with micro-OCT. And looking here at these stilled OCT video projections, where once again you have the filter, epithelium, and the ASL denoted by the yellow bar. We found no differences in air surface liquid depth or ASL between our control or SCI treated groups, which is quantified here, arguing that there was no difference in the hydration state of these two. But then looking at our mucus particle projections, where the severity of the angle of this arrow positively correlates to mucus transport. So the more angled this arrow, the faster mucus is moving, the less angled, the slower mucus is moving. We found that our STI treated group had significantly slower mucus transport, as quantified here, compared to our control. So this argues that independently of hydration, this salivary-transferase reduction resulted in more static mucus, resembling a more CF-like phenotype, and that this normal mucin saliation may be very important for normal mucociliary transport. So then we took it a step further and wanted to see if this held up in our in vivo model, which we use as a wild-type rat aged greater than 6 months. And we use these because they have mucus contributions from mucus-rich submucosal glands, which much more closely matches the airway microanatomy that you would see in the normal human. So we took our STI, we intratrachely instilled these in our wild-type rats daily for 7 days, excised their trachea, cut them open, so we could take those same cross-sectional videos to assess mucociliary physiology, and then we performed OCT on these again. And interestingly, our vivo data matched our in vitro data very well. Looking again, there was really no change in this ASL layer or the hydration state, and there was a significant decrease in mucociliary transport. And this is quantified here. So this supports that a decrease in saliation may result in a slower mucociliary transport phenotype, and reduced saliation may be contributing to this mucostasis that we see in CF. And this holds up even in an in vivo model with the contributions of mucus-rich submucosal glands. And so the conclusions I really want you to take away from this talk are that CF HBEs exhibit reduced mRNA levels of multiple solid transferases that are potentially involved in mucin biogenesis, as well as decreased alpha-2,3 and alpha-2,6 mucin saliation in CF is partially restored with elexacaftor, tezacaftor, ivacaftor modulator therapy. And that treatment of non-CF HBEs with this SCI reduces saliation of mucins and induces a CF-like mucus transport phenotype. And that inhibiting saliation in these non-CF HBEs in rat trachea impairs mucus transport without affecting mucus hydration. And the overall takeaway is that saliation really warrants further investigation as a mechanism of CF mucostasis and may be a potential therapeutic target. So I'd like to acknowledge and thank my mentors, Dr. Stephen Rowe and Dr. Jared Barnes, as well as the Stephen Rowe Lab and the Jared Barnes Lab and my thesis committee. And I'd also like to acknowledge my funding that's provided the means for me to perform the work that you've seen today. At this time, I'd like to take any questions, and I'd be happy to hear any thoughts that any of you may have. And I've enjoyed presenting.
Thank you, Alex, for these very interesting insights into mucin cellulation. One question to begin with from my side: what do you think? Why we need salicylic acid on mucins, and how does that help to improve mucus rheology?
Yeah, it's a great question. So saliation of mucin offers, a lot of the negative charges dispersed all across the mucin backbone, which is a huge factor in the repulsive forces that help mucin linearize when it's secreted, so it can take on a more healthy, expansive conformation. And it's also really important in all of the positive cationic interactions between, such as calcium and sodium, and that exchange when mucin is secreted into the airway. And so when these negative charges are altered, you'd expect that their interactions with other positive elements, as well as their repulsion forces, would be dysregulated, which is kind of the premise behind why we believe they're very important to study.
Thank you. And next question, any thoughts on the mechanism for the link between reduced CFTR and reduced cellular transferase activity or expression?
Yeah, that's a great question too. That's one that I knew would be asked. So there are a couple main dogmas where people think that salivation may be decreased in CF, and one of them that's been controversial over the years is organellular acidification due to defective chloride transport. So you have a lower pH within the Golgi where the salivtransferases will be working, and they're very pH sensitive, and so that would alter activity, but doesn't really give a reason of why expression would be changed. And so if expression is changed, really the strongest evidence we have right now would be possible CFTR trafficking defects that can cause a lot of organellular stress and initiate pathways such as ERAD and other things that may induce downregulation of some of these genes. So that's something we're definitely looking into. It's a good question.
Thank you. Then there are some technical questions. So when you treated with STI, your non-CF cells, you waited 24 hours before assessing transport. Was the existing mucus layer washed off prior to performing this experiment? And the second is, what's the mucin secretion? Was the mucin secretion rate or ion transport altered by your STIs?
Yeah, so that's a great question too. And so we definitely washed mucus off, and something interesting. So mucin, the salivation happens before intracellular packing. And so if we, treat it with our STI and then assess immediately, we would be looking at mucin that was already packed, that it wasn't really, didn't have any knockdown of any salivation. And so we actually try to express the release of these old mucin granules with UTP, and then wait a little while, wash all the old mucus off, and then treat with our STI. And so that any mucus that we're looking at would be newly synthesized mucus under these STI conditions. And we've also looked at other time points as well, but we had, enough of an effect at 24 hours that, range through at 48 and 72 hours as well. And as far as the ion transport changes, we haven't looked into that yet, but that is also something that we're planning on delving more into to see if we can identify any changes in ion transport due to this salivtransferase inhibitor. So.
Do you think it could also affect cilia beating or cilia?
Yeah, so that's something else we also can do with our micro OCT. We can actually calculate ciliary beating frequency to see how fast cilia are beating. And I did that, but it didn't show anything significant. And so we didn't really see any repeatable or significant changes in any ciliary beating before or after this treatment. So, but when this is published, that'll definitely be in the paper. So.
This micro OCT is great. You can have a readout on ASL height and the mucus transport as well as CBF rates. That's something I have to ask you later on, what you need to be able to perform this.
Yeah, there are some great posters too from some of our collaborators as well as us too, because I mean, there's a ton of things you can do that are still being developed on the machine. So it's a very interesting tool.
And definitely look into that. Thank you. Another mechanistic question. Do you think there are other conditions where this silation issue may be playing a role, like asthma or COPD?
Yeah, so if you remember some of my slides before, some of the premise for this is actually looking at muco-obstructive diseases in COPD or asthma, because there's been a correlation of this lower charge glycoform of MUC5B that have been implicated and found to be in higher abundance in COPD or asthma. And so, I mean, this is definitely something that could be more universal to mucus itself rather than just in CF, which I think is a really interesting concept. So.
Another thought-provoking question, does silation status impact the ability of bacteria to interact with mucins? I know, I assume you didn't look into it, but is there any literature data that would suggest so?
Yeah, that's huge. So, I don't know how much everyone here knows about antigens and host pathogen interaction, but there are, a lot of antigens such as the SIL-T antigen and such, where there's a specific ratio of fucose and salic acid that are very, very important for, I believe it's Pseudomonas recognition and such. And so alterations in the salivation profile would definitely be expected to alter host pathogen interactions. Exactly how, not quite sure, but it's definitely an interesting concept. Once we have the in vitro stuff, to take this a step further and look at it under infection models as well. So.
Great, because that was also a follow-up question if it could affect the clearance of pathogens, your STI treatment. And then a technical question, were you able to compare the amount of mucus in your different experimental conditions so that you could normalize against the amount you could recover in case there are differences in adherence or in case STI interfered with secretion? So you already mentioned that you used the UTP beforehand, right?
Yes. To
remove already synthesized mucins granules.
Yeah. So to answer that, sorry, can you repeat the question one more time? I got it.
Sure. Were you able to compare the amount of mucus?
Okay, yeah. So I got you. So we definitely, when we took this, we partially purified this in some size exclusion columns as well, and then normalized the amount of protein and things like that loaded, and what we worked with, and you have done mucin blots as well to ensure that we're using the same amount of mucin as in each condition. Because obviously, if you have more mucin that you're loading in one versus the other, then you'll have more saliation, and it'll throw your readings off. And so, yeah, we're trying to make sure we control for that very well. That answers the question.
I hope so. Have you ever tried the acute treatment of secreted mucin with exogenous neuraminidase instead of STIs?
That's a great question too, and that's something that I am currently doing. So, yeah.
But there have been what do you expect?
So that would kind of mention the question. Is it more of a mucin maturation issue you have? If you have decreased saliation, is it something as soon as the mucin's released and tries to expand? Is this where the saliation defect could contribute most? Or even if you knocked off the saliation, post secretion and post maturation, could this still induce a irregular mucus transport phenotype? And so I really don't have enough data yet to really comment on what I'm seeing with that. That's definitely something that we're interested in and are currently trying. So it's a great point.
Okay, fantastic. Then maybe one last question from my side. So this is N-linked glycosylation, right? But what about O-linked O-glycosylation?
Yeah, so the STI would really hit. So we really hit any saliation. But when we're focusing on looking at cell transferase levels and such, we're focusing on O-linked because that's where the vast majority of mucin saliation occurs, is through O-linked processes. So that's really the focus on this. And most of the data that you've seen is from O-linked glycosylation. So.
Okay, thank you very much, Alex, for this great discussion as well. Then let's move on to our next presenter, Dr. Divya Kamath from University of Kansas Medical Center, and she will be presenting an exciting new opportunity to modulate airway surface hydration.
Hello, everyone. This is Divya Kamath from University of Kansas Medical Center. Today, I'm going to talk about our work on role of methylthioadenosine in maintaining airway hydration in human bronchial epithelial cells. I have no relations to disclose regarding related to this presentation. As we know that CF is a monogenic disorder, but the severity of this disease varies even amongst people who have the delta F508 homozygous mutations. Recent genome-wide association study has indicated non-CFTR gene modifiers can have impact on the lung disease severity. Along the same lines, our nasal mucosal transcriptomic study from 134 people with CF has shown that there are differential gene expression in multiple different pathways can affect lung disease severity. Methionine salvage pathway is one such pathway that has been shown to be implicated in influencing the lung disease severity. Genes of this pathway, increased expression of genes in this pathway like the AMD1, the MTAP, MATTAP, MAT2B were associated with worse lung disease severity. MTAP is methylthioadenosine phosphorylase, catabolizes MTA or methylthioadenosine into methionine and adenine. MTA is the key metabolite of this pathway and is ubiquitous in eukaryotes. Preliminary metabolomic study has shown that lower amount of MTA in bronchoalveolar lavage fluid in people with CF correlates with worse lung disease severity. Also, MTA has shown to be anti-inflammatory in multiple models, including the mouse sepsis model, the DSS-induced colitis, as well as multiple different cell lines. Considering impaired mucociliary clearance as well as uncontrolled inflammation as part of cystic fibrosis, we wanted to see if MTA can not just ameliorate inflammation, but may also have a role in mucociliary clearance. Although we have studies which are underway for understanding the role of MTA on airway inflammation and airway physiology, today I'm going to talk about our work on the role of MTA in mucociliary clearance. So for this, we are using normal human bronchial epithelial cells as our model, and we are using TGF-beta as the stimulant. TGF-beta has been demonstrated to be associated with worse lung disease severity in vitro in clinical setting, whereas in vitro, it is able to reduce the airway surface liquid, ciliary beat frequency, as well as mucociliary transport by taking down the CFTR as well as the BK ion channel activity. As we can see over here, we have TGF-beta in our model that can take down the airway surface liquid volume, and MTA is able to restore it to DMSO levels. The ASL or airway surface liquid volume was measured using the meniscus scanning method. Similarly, we also see a restoration of the ciliary beat frequency as measured by Sisson-Amons, the video analysis method. And as can be clearly seen in these videos, TGF-beta takes down the ciliary beat frequency, whereas MTA is able to restore it. TGF-beta is also known to increase in expression of MUC5AC and MUC5B. And we see that in our model too, we can see an increase in MUC5AC and MUC5B as demonstrated by immunofluorescence staining. And this is taken down to levels similar to control by treatment with MTA. Consequently, we also tested the difference in mucus solids or mucus hyperconcentration demonstrated as percent mucus solids. We see that MTA is able to restore or reduce TGF-beta induced increase in mucus hyperconcentration. Considering that MTA is able to take down mucin expression as well as mucus solids, we wanted to see if it affects the viscosity, which is also likely due to increased in airway surface liquid volume. And we used the FRAP method or fluorescence recovery after photo-bleaching method to determine the relative mucus viscosity. The data is shown in the form of T half-life. Higher the T half-life indicates more viscosity, lower T half-life indicates lower viscosity. And as we can see that TGF-beta increases significantly, increases the relative mucus viscosity, and MTA it is able to get the viscosity down. Airway surface liquid hydration, ciliary beat frequency, mucus concentration, expression, as well as the viscosity, all this influence the parameters of mucociliary transport. So to study this, we grew our normal human bronchial epithelial cells on modified transwell inserts where they had a silicone center to provide directionality to the transport. We used fluorescent beams and treated the cells, and the mucociliary transport, or the velocity, was determined by calculating the velocity of the fluorescence bead, traveling of the fluorescence bead. And as we can see in this video, TGF takes down the mucociliary transport to a negligible level, whereas MTA is able to restore it. And as can be seen in multiple different across multiple different samples, we see that MTA is able to restore the mucociliary transport. To understand the mechanism through which the airway hydration, which is the primary cause for improvement in ciliary beat frequency, as well as the mucociliary transport, we carried out Using chamber experiments to study the effect of MTA on the ion channels. We tested MTA for the sodium ion channel, ENaC, CFTR, and BK, as well as CACC, which is not shown over here. And we see that MTA is unable to rescue any of these ion channel conductance in our experimental setup, indicating a possibility of different ion channel or solute carrier that may be involved in improving the airway surface liquid hydration. We also have preliminary data in CF airway epithelial cells using the same model. TGF-induced reduction in airway surface liquid and ciliary beat frequency, and the role of MTA. And our preliminary data does suggest the possibility of improvement in ASL and CBF in CF bronchial epithelial cells. So in summary, our key findings indicate that MTA improves key aspects of airway epithelial innate host defense by restoring airway surface hydration, ciliary beat frequency, and mucociliary transport after TGF-beta1 studies. And our studies are the first to indicate the improvement of the innate host defense in airway epithelial cells by MTA. MTA does not rescue ENaC, CFTR, CACC, or BK ion channel activity, but the defined improvement in ASL and CBF suggests the role of other ion channels or solute carriers. MTA is an adenosine molecule with a methyl moiety, and it is known to act through the A to B receptor in multiple cell types. A to B, as we know, is a G protein receptor, which can potentially increase cyclic AMP or calcium signaling, which should potentially improve CFTR, CACC, or BK ion channel function. And since we do not see improvement in these ion channel, we are investigating what downstream effector pathways may be coming into play when we are treating the cells with A to B receptor. Treating the cells with MTA. And literature has shown that MTA, although it acts through the A to B receptor in multiple cell types, it has different downstream effectors which are specific to the cell types. So looking into the downstream effector pathways in airway epithelial cells is crucial in identifying the mechanism of action for MTA. We also have other plans with MTA and CF related experiments. And as mentioned, one of the experiments underway is to determine the effect of mucociliary transport parameters in CF bronchial epithelial cells. And also, as mentioned in the initial slides, MTA is known for its anti-inflammatory properties in multiple different cell models, but its role in airway inflammation is not explored yet. And we are going to test the anti-inflammatory mechanism of MTA in paired HPBMC and nasal samples from CF patients. We're also excited to study MTDIA, which is a small molecule inhibitor of methylthioadenosine phosphorylase, or MTAP, which is the enzyme that catabolizes MTA. So when we use this molecule, it can stop MTAP from catabolizing MTA, leading to an intracellular increase in MTA level. Preliminary data has shown that mTDIA is able to increase bronchoalveolar lavage fluid MTA in mice over a 3-day treatment. The goal for studying mTDIA is to be able to develop it into a therapeutic application. That would be all. With that, I would like to acknowledge Dr. Deepika Paulineni, the PI of the lab, along with our lab mates, Morgan, Aidan, Ann, and Ted. A lot of this work was also done in collaboration with Dr. Salate's lab, and I would like to thank Dr. Salate and his lab members. I would also like to extend our thanks to Wanda O'Neill, Dr. Esther, as well as Dr. Knowles, as well as the funding sources, that is CFF Postdoctoral Award, BRTP, KUMC BRTP, and the COBRI Award. With that, thank you for your time and thank you for listening. Have a good rest of the day.
Thank you, Dr. Kamat, for the interesting presentation. And I think it was a very beautiful example of how an omics approach can identify novel therapeutic pathways, which could be validated then later on in cellular assays. Let's start with the questions. One of the questions is if you looked at other metabolites on the methionine salvage pathway?
So we looked at different metabolites because I know S-adenosine methionine, which is part of the same pathway, has previously in literature shown effects on ASL and mucus. So we looked at S-adenosine methionine as well as the polyamines through mass spec, and we see that at 24 hours, we do not see any change in the absorption or metabolism of this any of these metabolites compared to MTA. So for MTA under mass spec, we see that there is an increase intracellular level after 24 hours, which is our endpoint time that we use. Whereas for other metabolites, we do not see that change, and that's why we feel that the effects are through MTA.
And thank you. Is this MTA, does this have anything to do with ATP and adenosine pathway? So ATP that's secreted in response to viscoelastic mechanosensing and then metabolized to adenosine, or are they just name relatives?
So they do, MTA, when it is catabolized, it does form adenine, and then it replenishes AMP and adenosine. But with respect to the viscoelastic property, I don't think there is a relation to production of MTA with respect to any of the viscoelastic, viscomechanical or elastic properties.
Another question, if you looked at the effect of MTA on TGF-beta1 mediated changes in mucin secretion rates?
So we did do mucus expression as well as mucus solids. So those are the two parameters that we tested. We didn't do anything with respect to specific rates of secretion. We just did the endpoint of total expression as well as total amount of mucus solids, compare TGF as well as TGF plus MTA. And we do see a reduction compared to the TGF-beta does increase the mucus expression as well as mucus solids, and MTA is able to get it down to the baseline or control levels.
Some mechanistic questions towards what could be the mechanism of MTA. So have you looked at maybe ASL pH, if it would be a bit more alkaline, or did MTA application affect resistance, which would be indicative of maybe paracellular secretion?
That's a great question. Even we initially, once we could not figure out the ion channel, the known ion channels that impacted the ASL hydration, we looked at toxicity as well as we looked at the tear resistance, and it did not change the toxicity or the tear resistance. And that's a great point about ASL-PH. And recently, we have been thinking about looking at ASL-PH just mainly as even a guideline for just starting our investigation on which other ion channel we can work on. So no, we haven't looked at the ASL-PH yet, but we have plans to do that.
Then another question, your increases in mucus concentration in presence of TGF-beta1 are small relative to what observed in CF. Were these cultures washed at the start of the treatment, minus 24 hours, and have you ever looked at the effect of NTA after longer exposures to TGF-beta1?
So the first part of the question is, the mucus concentrations are lower compared to CF, and that is correct. These cells, which I've shown the data for, is for normal cells. They are not CF cells, but we do have CF cells ongoing, and we are looking at the mucus solids and mucus concentrations, and we do see a relatively higher percentage of mucus solids in that. So for the other part of the question, if they were washed, we do wash our cells 24 hours prior to treatment.
And another, if you ever looked at the longer incubation with the TGF-beta1?
We have tried 48 hours in the beginning, but we did not pursue that because 24 hours was giving us optimal stimulation and recovery.
So maybe I missed that, but is MTA extracellular secreted, and that's how it exerts its effect, or is it also intracellular? So how does MTA work?
So it acts, from literature at least, in other cell types. We don't know about airway epithelial cells, but it is working through the A to B receptor. So it can also be excreted outside the cell, if I'm understanding the question correctly. It is also present intracellularly. So there are, we ourselves have, our lab has mass spec data analyzed in patients for intracellular as well as extracellular concentrations, which is underway. Whereas in a cell in vitro setting, it can be, it is definitely present intracellularly in different concentrations depending on the tissue, and in certain cases it can also be excreted out of the cell.
Wonderful, thank you. I think we answered the questions that came so far. How stable is MTA? So do you have to reapply it, or do you know anything about the stability?
We haven't specifically tested the stability, but we don't have to reapply it. We treat TGF-beta as well as MTA at 0 hour and then take the reading at 24 hours, and we see the effect at 24 hours without reapplying MTA.
And your compound, this MTDIA, is it an experimental compound, or is it something in clinical development?
It has been tested preclinically and in mice and rat studies. Now it is commercially available at any manufacturer, and we can use it and test it. But it has not been studied further. It is in the initial phase of studies.
Another question is MTA altered in other diseases beside CF?
MTA has been studied in multiple different models. It is done in a lot of inflammatory disorders as well as in cancer. A few types of cancer where there is MTAP deletion, the enzyme that metabolizes MTA. So there have been studies of MTA with respect to cancer and other inflammatory disorders, like sepsis model, and even it has been even preclinically tested in multiple sclerosis. There have been studies for that very recently for multiple sclerosis.
Thank you very much. In case there are no further questions, then thank you again for your presentation and the lovely discussion. And then it's time to move on to our last talk from Dr. Feng Yuan from University of Iowa, and we'll be talking about a new in vivo research in ionocyte biology.
I'm Feng Yuan from University of Iowa, Anatomy and Cell Biology Department, John Ungerhall Laboratory. Today, my topic is generation of FOXL1 knockout ferrets using CRISPR-Cas9 gene editing to inform pulmonary ionocyte biology. Disclosure statement. There are no relationships to disclose related to this presentation. CFTR gene mutation causes cystic fibrosis, and what cells express CFTR gene? In a proximal airway epithelium is composed of different cell types, including basal cells, goblet cells, and ciliate cells, and so on. Each cell has its unique function. For example, basal cells are progenitor cells, and they are able to give rise to different cell types. And the goblet cells can secrete mucus, and the ciliate cells can perform mucociliary clearance. In 2018, there are two Nature papers came out, and both papers identified a new cell type called a pulmonary ionocyte. And both papers demonstrated that pulmonary ionocyte is the major resource of CFTR gene expression. And FOXL1 gene is the key transcriptional factor required for ionocyte specification and development. And both Nature papers demonstrated that FOXL1 positive ionocytes compose about 1% of the surface proximal airway epithelium, but express 50% of CFTR mRNA. So here we have the question, what are the functions of pulmonary ionocytes, and how can animal models help to address this question? Does ionocyte absence affect CFTR function? We started to use FOXL1 knockout mouse basal cells, our liquid interface differentiation system, to address this question. And during our collaboration with J. Rajagopal Laboratory, we found about 87% CFTR mRNA reduction in the FOXL1 knockout epithelium. And despite the CFTR gene reduction in the knockout epithelium, interestingly, we observed increased chloride current and bicarbonate current in the FOXL1 knockout mouse ALI cultures. And there's a fact, CF mice also express a CFTR-independent alternative chloride channel, which compensating for the dysfunctional CFTR. This may explain why CF mice don't develop cystic fibrosis non-phenotype. And a short conclusion from here, ionocytes are not required for anion transport in mouse trachea epithelium. The next question is this biology conserved in other species? Then we started to generate FOXL1 knockout ferrets using CRISPR-Cas9 gene editing tools. Two guide RNAs which target FOXL1 gene exon 1 has been validated by in vitro cleavage assay. Guide RNA is complex with Cas9 protein and injected into ferret zygotes. We successfully generated FOXL1 knockout founder ferrets, and those ferrets are harboring 5 unique indels. Then we started to bred those founders to get a FOXL1 knockout ferrets. And FOXL1 protein expression in the FOXL1 knockout ferrets was absent by Western blot, which confirmed we generated FOXL1 knockout ferrets. FOXL1 knockout ferrets showed kidney dysplasia phenotype. In 2018, Jason paper reported homozygous FOXL1 mutation patients, and those patients displayed kidney cyst by CT scan. And the FOXL1 knockout ferrets we generated showed a pretty similar renal cyst phenotype, and this is validated by CT scan and necropsy. And we also observed increased kidney body weight ratio in the FOXL1 knockout ferrets. And the FOXL1 knockout ferrets also showed kidney dysfunction, which is reflected by bone levels. FOXL1 is required for kidney intercalated cells differentiation, which is pretty similar to FOXI1 being required for ionocyte specification in airway epithelium. Then we started to evaluate alpha endothelial cells marker AE1, ADPase, and beta endothelial cells pendrin. The data we showed here is that AE1 expression and pendrin expression, ADP6V0D2, ADP6V1G3, and all the endothelial cells marker expression is nearly absent in the FOXI1 knockout kidneys, which is consistent with FOXI1 being required for endothelial cells differentiation. And ADP6V1G3 expression was absent in FOXI1 knockout kidney, which is confirmed by the immunofluorescence staining. Then we set up the kidney epithelial ALL culture and test the FOXI1 knockout kidney epithelial CFTR function. And what we observed here is FOXI1 knockout kidney epithelial showed decreased CFTR-mediated chloride currents and bicarbonate currents as well. A short summary on the kidney phenotype. FOXI1 knockout virus presented with kidney dysplasia likely due to the complete loss of endothelial cells. And FOXI1 knockout kidney epithelial exhibited decreased CFTR-mediated ion transport. And all the kidney data tell us we have been successfully generated FOXI1 knockout virus, and they showed FOXI1 gene loss of function. Then we started to switch gear to airway epithelium. We prepped proximal airway basal cells from FOXI1 knockout and wild type trachea and used this ALI differentiation cultural system to evaluate cell-specific gene expression and CFTR function. Cilia and mucin staining confirmed those ALIs are fully differentiated from both wild type and FOXI1 knockout group. And what we observe here is the key ionocyte marker FOXI1 gene, SL3, BSND, are nearly absent in the FOXI1 knockout airway epithelium. And this shows the absence of ionocytes in the FOXI1 knockout epithelium. And we also observed the CFTR mRNA expression was reduced in the FOXI1 knockout airway epithelium. And there is no change in other genes like FOXJ1 and the mucins. So FOXI1 knockout basal cells derived epithelia show absent ionocyte transcription factor expression and a decreased CFTR expression. And the FOXI1 gene knockout does not affect ciliated and goblet cells commitment. Then we started to test CFTR function by using electrophysiology. We set two conditions here. One condition is put the polarized wild type and FOXI1 knockout ALL culture in chloride buffer, which is bicarbonate free. And the other one is opposite. We put cultures in bicarbonate buffer, which is chloride free. And what we observe here is the FOXI1 knockout polarized ALL culture showed a decreased CFTR mediated chloride current and a bicarbonate current, as we summarize in the bar graph here. And there's a slightly change in the transepithelial resistance when the ALL culture is in chloride buffer. Summary on the electrophysiology data, FOXI1 knockout basal cells derived epithelia showed a 67% decreased CFTR dependent chloride current and 68% decreased CFTR dependent bicarbonate current. As we know, the L surface liquid ASL and its pH plays critical role in lung defense. So we are wondering whether absence of ionocyte may affect ASL height and pH regulation. So we have some experiments here. So firstly, we found those wild type cultures, they are able to respond to phosphocoline and IBMX stimulation. So when CFTR is activated, the wild type cultures are able to alkalinize the ASL pH. However, FOXI1 knockout airway epithelium showed abnormal pH regulation, as we show here. And we also observed the ASL height reduction in the FOXI1 knockout epithelium. So FOXI1 knockout epithelial cultures showed a lower ASL pH in the presence of phosphocoline and IBMX. And the ASL height was also reduced by 45% in FOXI1 knockout cultures as compared to wild type cultures. Then we're kind of thinking what new phylogenomic models can inform ionocyte functions. We created two genetic models. One is the Rosa TG Cre reporter, and the other is FOXI1 Cre ERT2. the Rosa TG Cre reporter virus is able to turn from TD tomato to EGFP upon Cre expression. And the FOXL1 Cre model is we inserted RCRT2 gene into the 3' UDR of the FOXL1 gene. Then we started to breed those two viruses together to get a double transgenic virus. And the lineage tracing of the double transgenic virus, Rosa TG and FOXL1 Cre viruses demonstrated that the tracing of kidney endothelial cells and ionocytes in the airway epithelium from trachea, bronchi, and the airway glands as well. And more interestingly, we found the pulmonary ionocyte was neighbored in the collecting ducts. So after we got the lineage tracing animal model, we are thinking, does pulmonary ionocyte responsible for directly transport ions? Highlight sensor has been described, can rapidly and quantitatively to measure cell function. And highlight sensor can be used as an iodide and chloride exchange indicator because highlight sensor can be quenched by iodide and show a higher sensitivity to iodide compared to chloride. So the idea we're presenting here is we selectively express highlight sensor into pulmonary ionocyte. When CFTR is activated, we add in the iodide into the apical site. The YFP positive ionocyte should be able to uptake iodide and secrete chloride. And this will make a YFP quenching phenotype. Then we generated a lentiviral vector, express highlight sensor, which converts to YFP upon Cre expression. Then we use a lentiviral to infect FOXL1 Cre basal cells, and that cells grow on ALL culture and under tamoxifen stimulation. We predicted to see the ionocyte being labeled by highlight sensor, and this is confirmed by lab cell imaging. Then we started to test CFTR function by using the highlight sensor method. We set up 3 different conditions here. One is chloride, one is iodide, and one is GH1 iodide. As the lab imaging show here, the iodide is able to quench the highlight sensor labeling pulmonary ionocytes. And this phenotype can be inhibited by a CFTR specific inhibitor, GH101. And relative YFP fluorescence intensity summarize all the data we collected. As the bar graph show here, the arrow indicates the different ion stimulation. And we can clearly see the highlight sensor labeled pulmonary ionocytes can be quenched by iodide when CFTR is activated. This phenotype can be partially inhibited by CFTR inhibitor GH101. And all those data tell us ionocytes are responsible for directly transport ions. So a summary for all my presentation. FOXL1 knockout virus present with kidney dysplasia, likely due to the complete loss of endothelial cells. And the FOXL1 knockout virus showed decreased CFTR expression in airway epithelial cell cultures, which is consistent with studies on FOXL1 deficient mice. And the FOXL1 knockout virus also showed decreased CFTR-dependent chloride and bicarbonate current. FOXL1 knockout epithelial cultures showed lower ASL pH in the presence of phosphocholine and IBMX. ASL height was also reduced by 45% in FOXL1 knockout cultures as compared to wild-type cultures. And most importantly, pulmonary ionocytes are responsible for directly transport ions. Acknowledgement, I would like to thank my mentor, John Engelhardt, for supporting the whole project and also for his super insightful mentorship. And thanks, Grace Gazer, for all the help on all those experiments. And thanks everyone from our ENAP. And a special thanks to my collaborator, Daniel Montorio, and J. Rajagopal. They provided us the FOXL1 knockout mouse basal cells. And also thanks Peter Heige and Alan Workman on the discussion of highlight sensor experiments. That's all my presentation. I'll be happy to take questions.
Thank you, Dr. Yuan, for this very elegant study to dissect the role of ionocytes in vivo and in vitro. Then let's get to the questions. One of the question which CRISPR-Cas9 sgRNA transfection method was used to generate the FOXI1 knockout ferret model?
Yeah, that's a good question. So the method we use here is an RNP method. We are complexing the Cas9 protein and the guide RNA. So it's a protein guide RNA complex, and do a zygote injection on that.
Thank you. Another question, in your electrophysiology data with the knockout cultures, you show reduced amyloid sensitive current. Any idea why? Do ionocytes have a higher INAC in addition to CFTR maybe?
Yeah, that's a good question. So actually, we have some experiment going on. So we're trying to single cell a lot of ionocytes. So some of the data I haven't shown here is that the ionocyte also express pretty high like INAC channel gamma. So maybe this can explain why we also observe the decreased amyloid sensitive currents.
I see. Thank you. Another question, do these ferrets exhibit a pulmonary phenotype?
Yeah, I think this is a question most people will be curious about. So all the FOXI1 knockout ferrets, all the data I showed here is from pretty young ferrets because they have pretty severe kidney disease. Like I showed, FOXI1 is being required for kidney cell specification. So they have pretty severe kidney disease and they can't survive. So the animals I sacrificed here, they don't show any like typical CF lung phenotype here. But we have some ferrets, they are survived from the kidney disease. So hope those ferrets will give us an answer on this.
Thank you. Did it come as a surprise that you have such a multi-organ morbidity in this FOXI1 knockout? When we think about, you also had a mouse model. Did the mouse model exhibit a similar phenotype, except for the lung, of course?
Oh, for the lung, the mouse. So we've been collaborating with the Harvard side from Jerry Group lab. So the FOXI1 knockout mouse, they showed as I showed in a presentation, all the like tricky epithelial cells, they show increased cellular current, which we explain this is an alternative like compensatory mechanism in a mouse, and that's why the mouse don't have a CF disease. But in a FOXI1 knockout ferret, the phenotype is like opposite as we show here. They show a decrease like about more than 50% CFTR mediated, both correlated by cardiac current. So that's the difference between the mouse and the ferret. And the other thing is because the FOXI1 is being required for kidney cell specification. And the other thing we found is a little bit different here is the FOXI1 knockout ferret, they showed a kidney cyst phenotype, which I showed in the slides, is but pretty similar to the human. And the mouse don't have this kind of phenotype.
Then another question, if you looked at the effect on mucus transport rates in these animals, and if not, what is your prediction since there is still 30 to 40% forskolin stimulated current remaining?
Yeah, actually we're doing some mucus transport rates on this. So we've been collaborating with the UAB set. We're shipping some of the FOXI1 knockout trachea to them. We did some pretty similar experiment, like they showed from the microcity method, and some of the phenotype, like synapial frequency, it didn't change, and I think I can remember one thing changes the ASL height, and which is pretty consistent with our in vitro ALL cultures on this.
Do you know? Yes, please expand on this.
Yeah. So regarding to the mucus transport rates, so we're still having some experiment on that. So maybe next time we can bring it up to you guys. Yep.
Do you know if culture media conditions could affect relative numbers of ionocytes, either in ferret or human cultures?
Yeah. Actually, our eLab, we have I think one of my colleague is trying to figure out what like different cultural media can affect RNA specification. We trying to compare the like AIL culture and the USG media, and I cannot get a really solid conclusion on this, but I do say they do have some changes, but I wasn't sure it's reproducible or not.
Thank you. Are there other cell types in the lung that have altered abundance in the knockout?
Yeah, that's a good question. Actually, we're still working on some single cell experiment on the FOXL1 knockout culture and wild type culture. Actually, I don't believe other cell types are changed. As I showed in the slide, FOXL1 knockout didn't change like GABA cells and salinity cells commitment.
Thank you. Then have you maybe looked at the pancreas? Is there any phenotype in the pancreas? And if you didn't, then what would you expect?
Yeah, short answer is no. And from we have a lineage tracing animal. We're able to trace the analysis in the ectoferret model. We didn't see any analysis in the pancreas. And we didn't see the pancreas phenotype in all the animals we sacrificed.
Then have you looked at the activity of alternative chloride channels in your knockout culture?
Yeah, so actually we're working on that. So like I said, we're doing a single cell sequencing. So we should be able to tell we're trying to find all the chloride channels in like which is differentially expressed in a wild type and knockout. We're trying to figure out which one is which, and we're trying to compile cross different species like mouse and ferrets on that. So this is one of the goals we're working on right now.
Have you maybe functionally looked at it in the Using chamber by a purinergic stimulation to look at UTP responses, for instance?
Oh, that's a good question. Actually, I can try that. I haven't tried that. I haven't tried the UTP on the Using chambers. Yeah, that's a good suggestion. Thank you.
Please ask your questions. We have some time left, so I encourage everybody to submit questions. And do you treat these ferrets like CF ferrets? Do you get the same chow, and do you give antibiotics to the young animals, or how do you try to keep them alive and healthy?
I think the question is asking about ferret husbandry. It's a kind of different from CF animals because they don't. So right now, because those ferrets they have the head child problems because the inner ear issue. So sometimes they just cannot focus on spot. So we have to do like we have to help the ferret to grow. So we have to hand feed the ferrets just by using the animal like milk, and we didn't put any antibiotics inside. Yeah, but it can help the ferret survive.
Okay, then there are no further questions in the Q&A. Then I would like to thank you for your nice presentation and the discussion, and thank everybody who participated, and all the presenters and the audience to engage. I think we had a very lovely session. I think it was we had a lot of variety in our speakers, and I really hope that everybody learned something today. Then Brian, would you like to add something to that?
No, I think just thank you to the audience for joining us here. I wish we were doing it in person, and I hope we get to see everyone in person next year at the North American CF meeting. So hope everyone had a good meeting. Again, I'd just like to thank Anita for taking charge of this, so you don't have to see me the whole time. You can see her, and thank you to all the speakers. I think you guys did a fantastic job. So thank you.
Thank you, everybody. Goodbye.
Bye-bye, everyone.